Herkese merhabalar. Nano Open'a hoş geldiniz. Bugün Nano Open'da tıpta giyilebilir biyoelektronikler hakkında konuşmak üzere Doktor Onur Parlak konuğumuz olacak. Hoş geldiniz Onur Hocam. Sözü ve ekran paylaşımını kendisine devretmeden önce hakkında kısa bilgi aktarmak istiyorum. Doktor Onur Parlak, lisans ve yüksek lisans eğitimini İzmir Yüksek Teknoloji Enstitüsü'nde tamamladı. 2012-2015 yılları arasında İsveç'in Linköping Üniversitesi'nde biyoelektronik alanında çalışmalarda bulundu. Bu dönemde programlanabilir biyoelektronik yüzeyler ve iki boyutlu malzemelerin biyosensör uygulamalarında uzmanlaştı. Doktorasını aldıktan sonra bilimsel çalışmaları ve gelecek vadeden bilim insanlarını destekleyen Avrupa'nın önde gelen vakıflarından Wallenberg Vakfı'nın desteğiyle daha önce yürüttüğü projeleri gerçekleştirmek üzere Amerika Birleşik Devletleri'ne gitti. Amerika'da Stanford Üniversitesi Malzeme Bilimi ve Mühendisliği bölümünde çalışmalarına başladı. Burada giyilebilir biyoelektronik cihazları üzerine yaklaşık 3 yıl çalıştı. Stanford sonrası geliştirdiği medikal cihazların tıbbi uygulamalarını hayata geçirebilmek için 2019'da İsveç'te Nobel Tıp Ödülleri'ne de ev sahipliği yapan Karolinska Enstitüsü'nde çalışmalarına başladı. Yakın zamanda Karolinska Enstitüsü tarafından olağanüstü bilimsel değerlere ve gelecek potansiyeline sahip önde gelen genç araştırmacılarından biri olarak seçildi. Bunun yanı sıra Avrupa İnovasyon Konsülü ve İsveç Araştırma Enstitüsü tarafından proje destekleri kazanarak 2021 yılı itibariyle de yardımcı doçent olarak kendi laboratuvarını ve çalışma grubunu kurdu. Doktor Onur Parlak'ın 30'a yakın bilimsel makaleye ek olarak giyilebilir ve programlanabilir biyoelektronik üzerine 2 tane de kitabı bulunuyor. Hocam hoş geldiniz. Sunum paydaşları ve sunum araştırmacıları giyilebilir biyoelektronikler
konusunda ilgili 2 tane makale başladı.
Pardon bir ses. Giyilebilir elektronikler üzerine tecrübeliler. Bizim de sunum liderliğinde yürütülmekte olan TÜBİTAK 1004 destekli Nanosys platformumuzda benzer bir konu işleniyor. Nanosys sağlık için takip ve tanıya yönelik hızlı ekonomik ve özgün nanoteknolojik bileşen ürün ve sistemlerin geliştirilmesini hedefliyor. Bunun 6 temasından bir tanesi de giyilebilir teknolojilerle sağlık takibine yönelik çalışmalar yapılması. Dolayısıyla bizim araştırmacılarımızın aşina olduğu bir konuda sizi de ağırlamaktan mutluyuz. Tekrar hoş geldiniz.
Hoş bulduk. Çok teşekkür ederim davetiniz için ve sunumunuz için. Ekran paylaşıyorum.
Tamamdır.
Şu anda görüyorsunuz umarım.
Görüyoruz ama tam ekran görmüyoruz.
Tam ekran yapıyorum. Tamam.
Tamam, harika.
Konuşmacılar arasında yabancı isimler gördüğüm için İngilizce devam edeceğim. Sizin için de bir mahsuru yoksa.
Tamamdır.
Çok teşekkürler. Thank you so much for having me here today. It's a really great pleasure to present my work on wearable bioelectronics in medicine. Today's topic will be mainly about wearable sensors and how we apply those sensors recently on the clinical and preclinical application, and how can we make meaningful application of these devices by collaborating with industry and with different clinical partner in Karolinska Institutet and out of Karolinska Institutet as well. So, but I would like to start the concept that we would like to emphasize here, and we see skin is the ultimate sensor. This is a kind of a motto of our lab, and I'm working in the dermatology department that we are always integrating the devices that we produce with human skin for health application. But before that, it's good to get a bit quick skin fact, important information about skin that I would like to emphasize here. So, first of all, human skin is quite one of the biggest organ in human body, an average of 2 meters square and about 3 to 5 kilogram. and is also may contain 600 sweat gland, which is another type of niche application that integrate wearable sensor for sweat sensing. In this respect, you have easy and quite a lot access to the sweat gland. And also, human skin is quite dynamic surface we are talking about, such as constantly human skin shed dead cell, 35,000 cells per minute, which equivalent of 4 kilograms per year. And also renew in every 28 days. It's part of dynamic property of the skin, and also quite hosting many different bacteria that are kind of part of the immune system, maybe we can call it, and protecting us against external the threats. But the concept that we want to do, as I said in the beginning, we would like to observe changes in the human skin and integrate devices and get some idea about the overall health and how our health can be monitored by integrating devices through human skin. But skin has many functions as well. As I said, there are many sensory receptors that helps us to interact with the environment. And also part of this, the skin microbiome and many other factors and the type of skin cell differentiation helps us to defend our body against environmental factors, such as the bacteria, different bacteria, which is not commensal, and also environmental conditions like UV light or the environmental pollution. But skin is also kind of a factory which produce so many different molecules just directly on the skin, independent from different body parts. And then such as vitamin D is one of the good examples for this. And different than other organs that we don't look for the other organ, but is also there is social side. That is also skin as a tool for self-expression in many different cultures all around the world. So in this respect, we are talking really, really special organ. So about what we do and what people do all around the world, trying to make devices and integrate with human skin to continuously monitor the human machine. So there are many devices already available or already under development. You can see some examples here, but these are the recent examples. We can find many more examples as well. And what we try to do also, take this concept of wearable sensor in the context of precision medicine. What we mean with precision medicine, this is also another branch of science, which is quite well developed, but we want to integrate the wearable sensor with human body, as well as the environmental settings that we are living in, to gather the information about our health, such as environmental, behavioral, physiological, and biochemical. With this way, we are aiming to predict, prevent first, but also predict any future outcome, whether it can happen in our body or not. So for this purpose, our lab focusing on different applications, starting from my early research in sport medicine. And nowadays, we have a couple core applications like wound healing and epilepsy prevention and prediction, but also by collaboration, we are also doing melanoma screening, preterm infant monitoring, as well as some skin conditions, skin diseases, by using the devices that we produce. But now I will take you a bit 10 years ago, the story about these things during my PhD research. I'm not going to go into detail, but as a successful PhD student, but also failed Ironman competitor, I had a question in my mind, then how, even though I'm well prepared, how I can fail, what is happening in my body during intensive sport activity. When I researched those things, one of the important factors was the stress. How the stress play a role for us to affect our performance during the sport activity, and how can we manage this? How can we get rid of this stress? But my research was to monitor the stress process during the performance monitoring. For this purpose, I wrote the project, as you mentioned in the beginning, and I went to US and do the research on designing wearable cortisol sensor. Cortisol is one of the indicators for the stress. Of course, stress is more complicated process. I agree with that. But we just decided to do the cortisol sensor and to monitor how our body cortisol level changes before, during, and after heavy sport activity. I'm not going to go into detail this work because they're relatively old. In 2018 and 2019, we published some work about that. But after that, even though we think that is a very nice concept for sport medicine, but over the years, our collaboration and different interacting with different group and different people, and now we are trying to expand this concept for the mental health and epilepsy research and monitoring the livestock to increase the yield of the animal production, for example, or also effect of hormones and cortisol, particularly in dermatology and skin research. So, and also quick look what we do now and how things started 5 years ago and how we're translating these devices now to make more independent devices. What I mean, as you see here on the left side, this is very simple device. This is just OECD devices. There is no electronic integrated. We always dependent on the devices in our lab. We need to go without taking our body, these devices. We need to connect them to the device and do the measurement. But in clinical perspective, this is not realistic. We cannot ask patients to come to our lab every 2 hours, every 4 hours, and do some measurement, right? So what we need to do is to design the device. It can monitor your movement, understand the daylight and the daylight conditions, whether it's dark or there is light outside, and also other capacity, not only one single molecule, but different molecules, physical and environmental things at the same time. So this concept even developing more nowadays. We are in that stage, but it's also we have very simple prototype that we are trying to do. With this way, we are not anymore dependent on the electronic devices. We have microcontroller, a small potential start integrated in this device, as well as battery, other electronic component as well. So as I said, the initial concept that we generated back in 5 years ago, now going to different applications and different the collaboration start over the year. And 2022, we start our European Innovation Council grant, the Council project with Neuroscience, together with other partners that we are doing, the collaborating for this developing sensor devices for sudden unexpected death in epilepsy prediction and populated prevention. In this case, I can go a bit detailed. We try to generate the concept, multiplex sensing concept, by integrating different the biomarkers, different indicators. But what we do, what I mean with this, that so everybody familiar with the glucose sensor, right? So this is type of sensor which is quite useful and is enough to be, as I told you here, one single biomarker, and you need to sense this biomarker. It's okay, but it's not okay for many other diseases. We need to do multiple parameters at the same time. So this type of device will not work in many different diseases. Again, let's take the glucose, and some of the patient group, you can, the critical conditions, you can monitor the glucose, but at the same time, you can inject the insulin onto the body. This is like artificial, the pancreas concept, that you need to have a closed loop system, which is solving the problem of instant elevation of the glucose. But what we try to do here, integrate this adapted sensor concept, which you monitor many parameters, biological, chemical, biochemical signal, as well as physical and environmental signal as well. And then you bring all this data together in collaboration. This is not the part that I do, but neural network analysis, artificial, the intelligence type, the measurement things that we bring and upload in our system. And then this helps us to predict and prevent the physiological conditions related to epilepsy. So this is the concept that we do, but also we added here a pump, means in case there is different conditions that you want to interfere with this, that you can also inject some drugs that helps to balance some of the biochemicals level in your body by kind of integrating sensor and drug delivery together. So another work that switched the gear a bit here, but talking a bit different concept that we tried to develop very recently, which is the real-time melanoma screening by designing microneedle sensors. Here is the important things that the melanoma, even though it's easy to diagnose, but there are certain conditions that differentiate skin melanosis from the different skin anomalies that we see in these pictures. For example, these are the melanoma example, but it's difficult to understand whether it is benign or not. So what we do for this, we try to design a microneedle sensor, single use, we design in this way, go to the place that you are suspecting that might be melanoma, and stay there 1 or 2 minutes, and then take out the sample, or when the sample, the microneedle on the body, you can measure many different parameters. So what we do, our target biomarker is here, tyrosinase. We have a microneedle, and we have special probe designed in our lab, and then tyrosinase interacting with this interacting with this smart probe, and then oxidized and reduced by the presence of tyrosinase. And we generate signal from the low to high concentration our device can understand. For this, what we do, the first step is to design a microneedle device, not the sensor at this stage, just the device. And what we follow is clean room-free microneedle fabrication with the conductive polymer combined with the polyurethane. So here we have a process, as you see here, we're generating pilot PSS-based microneedle, and then we are cutting them and separating them, working, countering reference electrode. And then we modify the reference electrode for the silver chloride in order to generate the electrochemical signal. And then we combine all this 3 electrode microneedle array and then modify the working electrode. As you see here in our working electrode, we have several steps. We are treating them with some chemicals, and at the end with the smart probe based on the L-DOPA. And then this L-DOPA oxidized and reduced by the presence of cortisol, the tyrosinase, and then generate the electrochemical signal. And then when you look at the microneedle under microscope that you can see here, this is a 6-to-6 probe, the device that we have, and a closer look to microneedle and another closer look to microneedle here. And also we do some insertion tests. We take these devices and insert in our skin for 10 times successively and recover it and do the SEM image analysis. And here also you can see after coating the pilot PSS microneedle with silver chloride, you can see there is any defect on the surface or not by using SEM. And we show that actually 10 successive insertion doesn't break and doesn't damage the pilot PSS and also silver coating as well. And then we do the performance analysis. First, we do everything ex situ outside. We kind of calibrate our devices and from low concentration to high concentration. And then after making sure that our device is really responding to different concentration of tyrosinase, and then we move to the ex vivo skin model. In this ex vivo skin model, we topically inject the tyrosinase into the medium that we know, that we control, and then we apply our devices, and then we measure under 2 different conditions, treated skin and untreated skin. As you see here, the treated skin means contains a physiological condition of tyrosinase, and untreated means there is no tyrosinase there, artificially added into the system. So, and then we can differentiate 2 different conditions at the same time. So, and then now I will move a bit different, the topics, different applications that we are applying our wearable sensor, which is will be about the wound and human skin wound. But as I mentioned, we have human skin and we have very well developed skin, and then protecting as many different environmental cues, including bacteria. But there are sometimes this defense system can fail. When this fails, many things can happen in our body, such as dermatitis, age-related with acne, psoriasis, And skin cancer is also a bit age-related, and also human skin wound. And these are the important, the kind of problems that our skin can face. And now we are dealing with those things, especially skin cancer and human skin wound and the psoriasis, and we are designing a device for these applications. So in the next couple slides, I'm going to show you human skin wound. So I try to blur the images, but it might be disturbing for many of you. Just the heads up before what is coming up. So if you are not happy with that, you can just close your eyes. But what we do, as I said, what's happened first, we have a human skin, and the defense fail means we are losing this tightly packed corneocyte, the bricks and mortar intracellular lipid matrix. And then our skin became more vulnerable, and many things, many pollutants, for example, can diffuse through, which cause such as psoriasis. Also, skin commensal and even opportunistic the bacteria can diffuse inside our body and cause infection. Here you see one of the typical example, very simple example. This is not bacterial infection related, but wound appear by age because exactly these things that I show you happen on our skin. So when the defense system of skin fail, it's losing lipid and losing the integrity, and it start to have the wound. But these are simple wound, but there are conditions that wound can go stronger and it can be more serious. So in our research, we try to design the sensor for two purposes. One is to the wound monitoring, how the wound infected by the bacteria and how our body deal with that. And the second thing is also we are collaborating with different people, with the clinician, to monitor skin biosis and how the changes of skin biosis affecting the skin diseases such as psoriasis. So this is another important things that we do. But I'm going to go into detail for the wound research. So as I said, the wound is very common in human body. And there are different type of wound. Mostly, one of the most important the things about the wound, the type of the wound that appear on our body is the diabetic foot ulcer. This is a bit for the problem people suffering from the diabetes, and once they have a wound, it's not healed very long time. And another important thing is the surgical wound that you have a surgery, and after surgery it takes so much time to wound to heal. Therefore, those two conditions is very threatening for the skin wound infection. So there are a couple stages of the wound infection. The first, let's look at here the acute wound, the wound that appear and close, quickly close. It takes 2 to 3 weeks, depends on age, it depends on your health. But the first case in the coagulation happen and then inflammation start. And the important process that we can monitor is the cell proliferation and the cell migration, and then remodeling state. But when you have the chronic wound, such as diabetic foot ulcer, and the wound happen because of the surgical condition, this process, the inflammation to cell proliferation, it really extends very long time. And it takes so much time to remodel the skin back again. So when you design the sensor, it's the right time to interfere, to understand is actually the process, the second step, the third step. You can integrate your devices and understand how your body reacting to the wound healing and what type of changes occur on human skin wound. So for example, I talk about surgical wound, and here you see how surgical wound heal over time. So here is very first day after the surgery, and it's not closing easily. And then over time, getting closer, and closer. But even the 70 days pass, still you can see the wound area. This is really big, the problem for the potential infection, bacterial infection, especially if you stay a long time in hospital. And it's another problem of hospital-acquired infection, which you need to really closely monitor. And here, one of the non-healing surgical wound example, the tissue and how the slope can be seen directly on the skin surface. So, but as I said, there are some treatment, there are some discussion about that, how you call the infection, which point that you need to start to call this infection, because just contamination, colonization, or after that you call it infection or not. I'm not going to go into this detail, but mainly the things that happening. First, bacteria come attached to the wound site and start to colonize. Then in many cases, it formed the biofilm, which I'm going to talk soon, and then bacterial infection became, in many cases, irreversible. And then even though you treat with the antimicrobial, the things that your body is not reacting to that. So, for example, methicillin-resistant Staphylococcus aureus, one of the biggest problems. But why it's happening? How bacteria colonize and you become your skin become non-responsive to the treatment. So because they form kind of strong colony there, which we call the biofilm, because bacteria not only stay as a single bacteria, but they colonize together and they form extracellular matrix. They embed themselves in this extracellular matrix and then become more and more resistant. So, and today's understanding, the things that you need to do is this, the wound swab, and take these devices, the wound swab, and then send to the hospital. And depends on the hospital and depends on the bacteria that you're trying to detect, it can take 1 to 5 days, which is very important, such as, let's consider the sepsis, the condition that end up with a very sudden death. Then you need to really go into the method that can give you the quick evaluation. So you need to really quickly understand, is it infected or not, or if it is infected, how my body is dealing with that. So for this, we are also designing the ex vivo skin model. That important thing is here that we are using the full thickness skin, and we also use immunocompetent human skin. So what it means is immunocompetent, because if you just take the skin out of context, if you don't keep them in really nice and supporting growth medium, bacteria can grow there still. You can make the wound model there, but the body will not, the skin will not react like they are reacting in normal condition, normal human skin. So therefore, it's really important to keep immunocompetent system, immunocompetent model that we try to use here. And then this immunocompetent model help us to realistically design human skin wound. So here we have the procedure that we start with bacterial culture and create the acute wound on it, and then let them grow for a couple days, and then we do image analysis and also electrochemical analysis. Here you see one of the example that as you see. So we sometimes we have two approach. First, in order to understand really how the bacteria grow, how the biofilm form on the skin surface, by doing sampling. What we do here, we have very small device, interdigitated microelectrode, and then we taking this electrode, with collecting sample from the wound site time to time for every hour or every 12 hours, and see how the bacteria grow. For example, in one of the example that we just used the planar surface, not interdigitated electrode. So here, when we just infection start, when we inoculated the bacteria, and how they look like, and after 12 hours they start to colonize more, but after 24 hours, it's really tragic change happened here. It's really they produce the bacterial biofilm, and the bacteria embedded there, and it's really difficult to get rid of this biofilm anymore. So this is very initial concept that we do. But what we do now, sorry for this graph. So what we do here now is we try to make the wound patch to measure the bacterial load and immune response, and also different bacterial metabolite and how bacteria communicate with each other, for example, quorum sensing molecule. So for those, we have very simple initial prototype that we do all devices and everything integrated in the single sensor, the battery, Bluetooth, microcontroller unit, and we have a sensor as well. And we also here as a concept, it's not really active yet, but the concept that we try to build up here is to interface that we can monitor bacterial growth, but also human skin response. For example, how our body reacting to the bacterial infection, such as TNF-alpha level, IL-6, IL-8, and different biomarkers as well. And then with this application, when you do this measurement, you can go and monitor how things change over the 1 month, over the 2 weeks. Then you can, your doctor and your nurses can get a clear idea. So, as I said, important factors that we do here, the biofilm monitoring and human skin wound, that we try to build up the model here. So, as I said, biofilm is important things, important challenge, and doing diagnostic about the biofilm, how they attach to surface, how they grow from planktonic to biofilm form. So what we try to understand here, when bacteria, the biofilm here, the bound to the surface, and how much they produce there when they bind, and where they prefer to bind, how they grow, what are the different levels of bacterial biofilm show on the surface. So for those, we need to know, I will open all of them here together, we need to know how bacteria communicate with each other, how bacterial electron transfer happens, so we need to understand. So one thing that direct electron transfer is by surface protein. Bacteria has so many surface protein, can be oxidized and reduced and interact with the solution that you have, or in human skin wound, they can also interact the same way. But also they can produce, depends on bacteria, some nanowire. With this nanowire, bacteria can communicate with each other. The other things is the redox active molecule, or also quorum sensing molecule can go into this category, and again depends on the bacteria. And bacteria can secrete this molecule, and this way they can actually understand and synchronize the behavior together to form the biofilm or move the different part of the wound or different part of the surface that we're talking about. The other thing, also bacteria can adjust the ion concentration, which we call ionic signaling, especially the potassium signaling. They can, and again, this is also depends on bacteria. You really need to know which type of bacteria you're dealing with in order to develop your method. So for those, we developed very simple, the model. In this model, we use Salmonella because we had very good control between the wild type and the mutant strain. So in the wild type, as we know, they produce cellulose and curli as an extracellular matrix, but the mutant cell cannot produce any of those, neither cellulose nor curli. So by using the QCM method that we are using, that we're growing them by air-liquid interface, and then we monitor how much they are binding to the surface. So important things that we try to answer is the when and how much biofilm form. And the left graph here can tell us quite clearly actually when they start to form the biofilm. So when we start is the black curve is the cellulose and curli, the wild type, and the red one is the mutant. So when they grow them to parallel system, they grow pretty much same. The bacteria grow, we know it, we monitor different way, but they don't attach to the surface, therefore they don't change the mass on the QCM chip. So, but after 12, 16 to 18 hours, something start to change there significantly. As you see here, 18 hours, the wild types start to bind to the surface and start to reproduce extracellular matrix. And here the histogram graph that we can analyze at the end of 48 hours, how those changes happen. And there is very clear difference between them. And we see some binding here and the LB medium as well, because LB medium, this is kind of growth medium that contains so many different proteins and small molecules that can bind to the surface. And then what we do, so we need to compositional analyze how things change. So we get our surfaces and with the stain. We are using special stain that stain binding only to extracellular matrix. They are not binding to the bacteria. They only prefer extracellular matrix like carbohydrate there, like cellulose and purine. So we look two different places, and we also, of course, I should say that we are using bacteria, GFP express bacteria, that gives us the green color that you see here. So first 12 hours, 6 hours, we do analysis, but I summarize here starting from 18 because there was nothing before. And 18 hours, some biofilms start to appear. So as you see, here's a red color, it's come from the biofilm, and the green color is the bacteria. And 24 hour, 36 hour, and 48 hour, you can see that red color dominating the medium because more and more extracellular matrix produced. So of course, we do the control measurement. When you look at this confocal images, there is no change, even though we use exact same condition, but with the control strain, there is no significant change happened for 48 hours. That means there is no mass formation. So, and then we do another morphological analysis using the SEM microscopy. So it's also important finding here, there are 3 different regions on the same bacterial, the biofilm wild type bacteria, and 3 distinctive morphology you can observe. Here, very on top, the biofilm that have access to more nutrition and oxygen, which is the important factor, and the biofilms start to be more resistant on the surface. But when you go in the middle phase of this biofilm, they are densely packed, but they are not as porous and fibrous as the first layer. And when you go to the third, very bottom, that less oxygen availability, and you can see the different morphology of bacteria. They are not producing, maybe there are some, as you see here, very at the bottom, they are producing, but not as strong as first and the second level. This also gives us important information when the wound happen on the human skin, and they also very resistive, we assume, on top of the wound, because they have more oxygen and more nutrition there, and less interacting with the human body, of course, idealized here, and that means they are more free to grow and they are more free to secrete different molecules and extracellular matrix. So coming to the end of my talk, we also, as I mentioned, try to apply these things for clinical, preclinical, and industrial collaboration. So I'm not going to name any biomarkers here because we have agreement with the company, but we are also integrating different sensor devices, as you see here, very simple prototype, simple devices that can integrate it with, for example, a diaper, the adult diaper and baby diaper, to monitor the infection and different the skin diseases that can form because of these products. And it's also a quality check for the product, whether they are good or not, cause any skin irritation or not. We can measure with these devices. So that's another important collaboration. So before concluding my talk, I would like to give a couple the bullet points, take-home messages. So let's look at here the major and discuss the major technology requirement for the widespread use of wearable sensors. So here, very good example. We know that this is the glucose example, starting from 70s, 60s to today. We came to the wearable application of the sensor. But for many biomarkers, for many diseases, we are not even here. So, but why? Why it's happening? Because glucose has some special things. Enzyme is relatively easy to detect, and you have relatively stable biorecognition element, but it's not the case for many other. So what are the important requirement is missing thing for the other things, and that researcher focusing on nowadays and try to solve this thing is the specificity. So you can have something detect the molecules that you want to detect, but at the same time, you should be really specific to this molecule, blind to the other molecule. For example, I can give you many examples. Antibody. Yes, we know that antibody is very selective to the molecule of interest, target molecule, but in the context of human physiology, human skin, human body. But if you take this, the antibody, and put in electrochemical concept, electrochemical perspective, they may not always very specific to molecule of interest. So this is very critical step. The efficiency of sensing, repeatability, reproducibility of the sensor, also important, but the stability is the another thing. So let's go back to the antibody or aptamers or DNA, enzyme. These are the common biorecognition, synthetic or semi-synthetic, natural and semi-synthetic recognitions, but they might be very stable on human body, but they are not stable in many cases out of the human body context. So if you put them on the electrode surface or optical measurement, different surfaces, they may not be as stable as before. So that's very important step that we need to solve. So our lab is also focusing on designing. At some point, some of them successful, but mostly, to be honest, unsuccessful. It's very difficult to stabilize these molecules. So another thing, limiting step, but I would say that it's going way faster than the sensor research is the microelectronic, means integrating electronic circuitry for powering, data transfer, and also the particular sensing application. So it's not that limiting, but still important because you want to make a device. This device really should be small if you do wearable application or implantable application. So another thing, as I mentioned, for the glucose case, may not be the problem. But if you go for different diseases, then you need to measure many parameters at the same time. That means you need effective data analysis, which can be also provided by artificial intelligence methods such as machine learning and neural network analysis. You definitely need to include those parameters. These are two things: is electronic and the data analysis through the artificial intelligence. I would say that they are the future of the biosensory research. So before ending my talk, I would like to thank my team here. As you see, it's a bit changed over the year, but former members also there. And I would like to thank also the funding agency. But what I want to say here, I highlighted the bold, the Turkish students come to me over the years for summer research. So my lab is always open. Unfortunately, not everyone, but at least 1-2 students I would like to host from Turkey, and I always try to do, but not only Turkey, so for everyone, but of course, they are welcome. So you can always come and visit my lab. So you need to communicate a couple months before. And also I would like to thank our academic collaborator and clinical collaborator and industrial operator here, as you see, for one of the work that I present today, I did with Muhammed Üçüncü, help us for the XPS analysis, and Alexander Zin for the wound application, Peter for the other project, and Ding for the another wound project that we are collaborating, and also industrial collaborator. So thank you so much, and I would be very happy to answer your questions if there is any.
Thank you. We have a chat box. There are 2 questions. Can you read them?
Yes, let's go. Yeah. Yes, that's first question. Maybe I can read if not everyone has access. First thing is the silver. Penetrating the skin with the silver microneedle. It's important point. So we also discussed these things. The way, especially that we do the spray coating, is not stable. We are so much aware of this, and I am sure that silver can be leaked from this. But what we designed here is, as I said, is not continuous monitoring. It's just exposing this microneedle sensor to the skin, and measurement takes 30 seconds. But of course, altogether, maybe it can stay in the skin for 1 minute, not more than that, and we think that it's enough. But of course, you need to do all this testing as well. I agree. I hope it answered the question. The second, can we separate bacteria species among microbial population on wound by one wearable sensor, or we just need to optimize individual sensor for each bacteria? So what we try to do here, yes, is a very good question. So we know there are potential skin bacteria, the wound bacteria like that, Staphylococcus aureus, Pseudomonas aeruginosa, and a couple others. These are the most dangerous bacteria that we need to take care. So when we design the sensor, we try to do multiplex sensor. We don't target one single bacteria. We target multiple bacteria at the same time. At this stage, we target two, but of course, two might be enough in this case, such as Staphylococcus aureus is extremely important. So first thing that we need to look is that, and Pseudomonas, and I think it's good to start with. But of course, by integrating different electronics, shrink the size of electrode, you can target more. So that's what I think about this. And the other question, has there been any progress in your research on non-invasive wearable sensing device? So yes, what we do, we also, I didn't present today everything, but we also try to do just a planar sensor, not penetrating to the skin. So we try to monitor different molecules. That's a very important goal for us, to do non-invasive sensing rather than penetrating the skin with microneedle or different things. So yes, we do research on that as well. Hopefully we will have more information in the future. So I'm reading the other one. As I know, some of the nanomaterials have been used in medical healthcare, such as hepatides in artificial bone or sensor or marker. What do you think of nanomaterial usage in your studies? We don't use nanomaterial particularly, but as long as they are stable on the interfacing medium, at the sensing interface, I don't see any problem, because I also have a material science background, so chemistry and material science background. I also never understand people scare from chemical and scare from small material. I don't think that is a problem, but of course it should be stabilized. It shouldn't leak to the surface of the skin, because then while you're trying to treat something, you can create another problem which you don't want to do. And it's an important question that we always receive from our partners, clinical partners, that also they're very cautious about that. And this is definitely the limiting step as well. But stabilizing is the solution. You need to find a way. I don't want to skip any. Yeah, okay. Thanks for your presentation. I would like to ask you, are you any experience on wound healing model on in vitro skin model? Yes, that's what we try to do. So we have a skin model. In vitro, yeah. We don't work on in vitro skin model. We have ex vivo skin model. But there's also another important things. I know a couple of papers there. People work on in vitro system by growing, for example, keratinocyte, which is good. But in many cases, it's difficult to generate the human skin response. How you can do that by just doing keratinocyte model? If people find a way, of course, the mimic the human reaction, human response to the wound, then I think it's very nice way to do that. But we don't do it. Okay. Another question. Can we separate? Okay, this one we answered already. Okay. And I think this is the answer to that question. A sensor that can distinguish between the gram-negative and gram-positive bacteria. Yes, you can design your sensor to differentiate those things. Even sometimes you don't need to do sensor. There is, I know a couple of good examples that you call artificial tongue, that you do this and you can differentiate actually gram-positive and gram-negative bacteria without using specific biorecognition element. There are papers about that. Reliable paper. Do you have any experience carbon nanomaterial based sensor? What do you think they use? Yes, I use a lot actually. During my PhD, the graphene and molybdenum sulfate, tungsten sulfate based sensors. Nowadays I do polymer based, but definitely they are good because they're also nice interface for help you to keep the biorecognition element stable and amplify the signal compared to regular carbon and polymer electrode. It's definitely a good idea to use those. Yeah. But depends also how you modify it. Yeah. What is the penetration depth of microneedle in the skin and what material used for microneedle? So there are many different things, but for our research, what we do is 800 micrometer. Goes deep into the skin, which is passing to epidermis, but somewhere in the dermis. We are targeting. If you go more than 1.5 micrometer, then you start to go under the dermis. That there are specialized skin cells and neurons there that you can touch them and then create the pain. So it's better to keep it as short as possible, but long enough to go to the dermis. And again, depends on application. So another question, the question that I asked for already. So based on how do you separate them? Based on their virulence factor like antigens on the cell wall, or is there any way to do that? So we are using the bacteria, the biorecognition element, such as aptamers, for example, targeting only, for example, Staphylococcus aureus. And then we have another aptamer that targeting bacterial protein on the cell wall of vice versa, sorry. The aptamer targeting bacterial protein on the cell wall of Staphylococcus aureus, and aptamer targeting the Pseudomonas aeruginosa directly. So this is how we do. Polylactic acid base sensor, no, I haven't worked. I don't know very well. I mean, we did actually something, but they are more dissolvable microneedle, so we didn't advance on that. So I can say that no. Another question from Gizem. Could you elucidate the design? I can read it. Could you elucidate the design, consideration, and challenges involved in ensuring precise diagnosis alongside targeted and controlled drug administration? And how do you envision overcoming potential hurdles such as dose regulation and compatibility with verifying the skin type? Yeah, that's an excellent question. That's all depends how you design your sensor and how you program your sensor, because you want to release the drug, but first question, how to store the drug. That's many people don't talk about this in real application. They just, of course, lab is easy to inject the drug, but how you store the drug on the device. So the first challenge I see more before what you asked. The other things, of course, you need to really know the physiology, the pathology of the disease. So when do you need and how much do you need? All depends on the conditions. For example, maybe for bacteria, you can inject something. For example, the microbial things. Once you see the bacteria grow there, but like for epilepsy, for example, is a very big deal. How much drug you can release, and skin type, of course, important because all related to the diffusion. So how they will diffuse from epidermis to dermis is varied, depend on the skin type. So yeah, that you need to do really intensive research. Thank you. You gave example about integrating your sensor to adult diapers. Do you have any other field that you're integrating your sensor on it? Most skin, we are integrating our sensors so to do the monitoring. But as I said, so many people integrating with different daily clothes, watch, and other things. There is no limit for that as long as you get the constant and good signal. But we mostly interface with the skin directly or with the materials like diapers.
Ask a question.
Yes, I was about to reading, but you can ask, yes.
Yes, sure. Very nice presentation. Thank you for sharing. So I was wondering whether you know if there is a commercial device to measure stress.
Yes, there are devices. They are measuring skin conductance and different parameters such as heart rate and then sweating rate. There are commercial devices very recently appeared, so you can buy them. But
are they suitable for everyday use, or you need to like
yeah, they are suitable for everyday use, but as far as I know, I can't speak on behalf of company, but I think they are not ready for clinical applications, just for leisure activity, like smartwatches, similar to that one. Yeah.
Because back in like 2021, I was collaborating with Canada and Taiwan, and we were interested to check the accuracy of PPG sensors in the wearable devices to measure health parameters and also mental health. Well, there are limitations like lack of gold standard and limited population, etc. In case of stress, okay, there's PPG, like, and the most accurate PPG has Apple Watch has that. It measures via heart rate and heart rate variability.
Yeah.
But then again, there are like a bit drawbacks because sweat and the temperature of the skin gives you maybe better results.
Yeah.
Then you need to take out your watch and put it on your palm to measure the sweat and the temperature.
Exactly. Whether you believe or not, I'm wearing one of the smartwatches, and now it saves me time to relax. So it's really measured the heart rate and my movement and speech, probably. So frequency. So therefore, it takes you that you are stressful or not stressful. So there are very simple devices actually you can measure. But as I said, in order to make really clinical application, again, I cannot talk about their device because it's a commercial device, but it's difficult to go for clinical application. You need to really go chemical or physical, not physical, but chemical and biological parameters for diagnostic purpose. It's my opinion.
But then you need applications installed in your, most probably you have an application on your phone, which tells you about the stress. I have a couple of them. One of them doesn't really work, but the other one works pretty well, actually. So it gives you, when you're not sleeping well, gives you notifications that you're experiencing a high stress level and you need to relax a little bit.
Yeah. There are. I mean, this field really progressed very fast. So 10 years ago, there was nothing like that. It's just measuring the heart rate. But now, more smart system coming, not only heart rate, many other things at the same time. And then analyze all those things, again, based on the things that they create, interface. So, and then they tell you stress is high or not. So it's possible, of course.
Thank you very much.
Yeah, we are coming to the end. If there is more question I can take. Yeah, there is one question about the microneedle. Specific advantages, disadvantages of clean room free microneedle in comparison to traditional microneedle manufactured in the clean room. Could you provide insight into about clean room free microneedle consideration factors, performance, safety, overall efficiency? First, really most important things for us is very cheap. So we don't have to pay all this clean room access and the training and everything. So we are able to produce this microneedle in our lab. But of course, it may come with many drawbacks at the same time. I don't deny those things. For example, the stability we show 10 times, but maybe if we do 100 times, the device will be unuseful. So what we tried to do, there was the deal. So either you go for, we also do clean room microneedle fabrication, but in this particular one that I mentioned was to produce as cheap as possible. And with the materials that we have in our lab and without buying excessive, all this clean room material, masking, etching, and other things. So we just didn't use all of those things. So our main goal is the price. We produce very cheap compared to the clean room. But of course, we probably give up some of the stability, for example, I can say. Or, for example, there was another question about the silver, which is a very valid question. So I am 100% sure that silver may leak over time. So, but when you fabricate in the clean room, you can do electrodeposition. You can have stronger reference electrode for sure. So that's all correct. So I think that's all. Thank you so much. And you can always write me as well. I would be very happy to answer your question if you have any other. So.
Onur Hocam, thank you very much. It was a really great webinar. Thank you, dear participants. I hope to see you next week, another researcher and another topic. Greetings from Istanbul, Onur Hocam.
Thank you so much. And thanks everyone for joining. It was a pleasure. Bye-bye.
Goodbye.
Bye-bye.